All right, so good morning or good afternoon, depending on where you're joining us from, everyone. We have kids coming in from all over the world, both live and on YouTube, so welcome in as we continue to highlight and celebrate amazing scientists, explorers, and conservationists around the globe. So my name is Jesse. I'm with Exploring by the Seat of Your Pants, and we do 50-plus broadcasts every single month with amazing scientists, explorers, researchers, and amazing groups worldwide. It has been so nice to have you all tune in live and on YouTube in this craziest of years as we continue to celebrate these very, very cool people. So today we are continuing a series that we began just last week with the Gardner Foundation. So the Gardner Foundation celebrates and showcases and awards the top global health and biomedical researchers in the globe. It is a fantastic organization based right here in Toronto, where I am as well. Last week, we covered COVID and vaccine developments with Dr. Tanya Watts. A really, really cool presentation, and I encourage you to check it out on our YouTube channel. Today, we are continuing with something a little bit different and something that's very exciting, but that a lot of classes only have a little bit of an understanding of, and that is the CRISPR-Cas9 system. So you guys might have heard this in popular news articles. You might have seen this in books or magazines or articles. The point is, it is one of the coolest technologies in the entire world. It's one of the most cutting edge advancements in science to happen in decades. And we're going to learn from it from one of the top researchers in the world working on it today. So Dr. Barrangou is at North Carolina State University. He's going to talk to us today about CRISPR, gene editing, and food across the board. I'm so excited to have him, and I cannot wait for all your questions live and on YouTube. So without further ado, thank you so much for joining us, Dr. Barrangou, and take us away.
Well, I'm glad to be here. And again, good afternoon, good morning, maybe even good evening. I'm delighted to be here. There could be arguably no more timely, no more compelling, no more interesting technology than CRISPR. So some of you have heard about CRISPR, have seen CRISPR, maybe even have used or eaten CRISPR. And what I'm going to do today in the spirit of the Gardner Foundation to celebrate, convene, and inspire maybe even the next generation of scientists. Some of you are in the room here virtually, assuredly. I'm going to introduce you to CRISPR, tell you a bit about genome editing, and blow your mind with regards to how far we have gone and how globally impactful it has been and it is poised to be. So for the spirit of the Gardner Foundation and the spirit of the speaker series, this is about science. And science, notwithstanding the skepticism and anti-science bias that can occur in the world, is one of the most pleasing, one of the most mind-boggling, enjoyable, but also challenging disciplines throughout the world. And what we want to do is really bring science to the public. And there's a series of steps we have to go through. We have to turn science into technology, the science of CRISPR into genome editing technology, use it and apply it to generate products like therapies, but also foods and ag products, and eventually, hopefully, get it accepted by the public to make a real impact. And in order for that to happen, academics, real scientists in the virtual world of teaching and learning and research have to work with industry and government and society to ensure that the best science brings about the best technologies and that those technologies bring about the best products to make a real difference in the world. And there's no more timely nor perhaps more compelling, tied with vaccines perhaps in the COVID-19 pandemic era, topic or technology than CRISPR. This technology of 2020 Chemistry Nobel Prize for my friend Jennifer Doudna fame has taken the world by storm. And I'm not going to talk too much about data and metrics here, but to give you an idea, there's about 200,000 labs in the world across 100 different countries that have and are using CRISPR-based technology to edit the genomes of any organism pretty much you can think of. This technology is revolutionizing science, but also revolutionizing the industry in many different industries. It is explosive. This is the number of papers that have come out on the topic in the recent past. You can see from a handful just 20 years ago, albeit before some of you were born, all the way to thousands per year. It is very diversified in terms of how it's being used. I'll go through that in a minute. And it is very global. We can see pleasantly here Canada in the top 10 countries with the biggest appetite for CRISPR-Cas system from a research standpoint. And obviously the US, China, and Germany rounding the top 3. So what is CRISPR? Where does it come from? What does it do? How does it work? And also, how is it impacting our lives as consumers, our lives as patients, and maybe even our lives as future scientists. So I'm going to show you a short video of Human Nature, a feature documentary relating the story behind and the applications of and implications of CRISPR-based genome editing technologies.
Mother Nature gave us something that's richer than our imagination. We saw a very peculiar pattern.
Never seen anything like this before.
I remember him saying, remember this word, CRISPR.
You can actually use CRISPR to change DNA.
David's doctor told me, just hold on, there's something coming. We've never had the ability to change the fundamental chemical nature of who we are, and now we do. And what do we do with that?
Our pig is the most advanced genome-modified animal running on Earth.
My lab has been accused of taking science fiction and turning it into science fact. I consider that very high praise. We could engineer a single gene that could potentially make us all more muscular. But should we make that universally available?
Many people find what we're doing to be disturbing, or they're not ready for it.
Что человек может создавать человека, человек, который может воевать без страха и без боли. И вот то, о чем я сейчас сказал, может быть страшнее ядерной бомбы.
Should we really be manipulating the heredity of future generations given our lack of knowledge about so many things?
I don't know where you draw the line between not having albinism and deciding your kid needs to be an extra foot taller so they can be a good oarsman and go to Yale. Where is that line? Who's going to draw that?
Anything that would stop my child from suffering, I'm for. You know, draw this ethical line wherever you want, but don't draw it in front of my disease.
Humans are very good at inventing things, but they're very, very bad at working out what the implications are.
The balance of nature is built of a series of interrelationships between living things and between living things in their environment.
Often you don't realize that you're in the middle of a revolution until after the revolution has occurred.
So you just heard this. Often you don't realize you're in the middle of a revolution until after the revolution has occurred, right? And we are now witnessing in real time the CRISPR revolution. As you heard from my opening comments, my friend Jennifer Doudna and her colleague were recognized last month with the 2020 Nobel Prize in Chemistry for their discovery and development of CRISPR-based genome editing technology. And it's a two-step process that allows scientists to rewrite DNA, the code of life, virtually in any organism you can think of. If DNA were written in a Word document, it's the equivalent of hitting Control F and selecting very precisely in the book of life 3 billion letters of it, right? A very large book, very tall book, probably a couple of stories tall. We can go in and selectively find a location in the genome where we can use CRISPR to generate a cut. Think of it as a molecular scalpel for DNA surgery. And precisely at that site of cleavage, we can rewrite, correct, alter, or edit the DNA content in the DNA sequence at will. This powerful DNA genome editing technology has been used in the last 8 years by the 200,000 labs I've mentioned to edit the genomes of any organism you can think of. Model organisms at the bottom, simple organisms like viruses and bacteria, all the way to non-humanoid chips and plants. In the food and ag industry, any crop of interest to you, including vegetables and fruits, or any livestock and meats you eat. And then, of course, at the very top of the value pyramid, medicine. And as a matter of fact, the power of CRISPR can be illustrated as follows. The wing color pattern of butterflies is unique in the world. No two butterflies have the same wing pattern. Genetically, scientifically impossible unless they're clones. CRISPR is so powerful You can take some cells out of this butterfly right here, use CRISPR to target a gene called yellow, and grow a butterfly with the exact same wing pattern in which the color yellow has been removed. Think about this. You can use CRISPR to recolor the wing patterns of butterflies. If you can do this, I would argue there is no technical limitation to what we can do with CRISPR other than our scientific imagination. And the reason this is powerful is because this technology has many technical advantages, and I'll walk you through. It's the most promising, portable, convenient, and powerful technology to rewrite genome across the tree of life that we've never seen. There's a few caveats, there's a few opportunities for enhancement, there's a few technical challenges, but the bottom line is at this very point in time, it's good enough, it's sharp enough, it's useful enough, it's affordable enough to enable about 1 million scientists every day to alter DNA of species of interest to us across the tree of life and across industrial segments. And in the last 8 years, those CRISPR technologies have been put at work in the research community to really alter the genetic content of organisms using biotechnology and ag and medicine. Think of any bacterium, any yeast, any algae that are used in the food supply chain. Fermented foods, yogurt, cheese, but also detergents, enzymes, soaps, biofuel, bioenergy, and the like. The same in agriculture, plants, livestock, but also engineering the forest, engineering flowers, engineering Christmas trees, engineering aquaculture species like shrimp, or of course, therapeutics, right? The power of correcting genetic disease, generating vaccines, antivirals, engineering microbiomes, sequence-specific antibiotics, cell therapies, immunotherapies, on and on and on and on. And as a matter of fact, already, within 8 years, the first proof of concept that CRISPR works to edit human genomes in a tissue, in a dish, in a lab sample, in vitro, we already have in the US, and soon in a number of locations in the world, patients who have been treated with CRISPR-based gene therapies to correct genetic disease. If you're curious about this, watch Human Nature. You will hear the story of David Sanchez, a sickle cell patient. This can be applied to beta thalassemia, blindness, cancer, hemophilia, sickle cell, Leber, and on and on and on. Any genetic disease for which the gene has been identified and we have a delivery modality for, we could scientifically, technically, fundamentally correct and rectify and get rid of disease genetically in a patient. Now, besides the obvious gene therapy applications, there's a lot of potential in food and ag. As a matter of fact, CRISPR-Cas immune systems come from bacteria. And the first ever demonstration of the function of the CRISPR-Cas machinery was in a bacterium used broadly, internationally, throughout the globe to make yogurt, to ferment liquid milk into solid products or semi-solid products like yogurt and cheese. This is also applicable not just in fermented foods, but also foods and vegetables, right? Think like tomatoes right here. Think cheese. Think mushrooms, right? Also in animals like pigs and cattle. You can actually engineer strains of cattle that do not create horns, so you don't have to take the horns out and have a more humane supply chain. Obviously, this applies as well to crops, corn, wheat, soy, rice, things like lettuce, right? Even nuts, bananas, on and on and on. And then non-edible items like tobacco, cotton, hemp, trees to engineer a more sustainable forest, and obviously cellulose for biofuel. We can use CRISPR-based genome editing to alter and breed next generation products throughout the world and throughout the food supply chain. This also applies to editing microbes, bacteria, microbiomes that they constitute, and you can think of engineering ocean microbiome, the plant microbiome, the food microbiome, the livestock microbiome, the soil microbiome, the air microbiome, and on and on and on, and all the different microbiomes associated with humans, the oral, the gut, vaginal, the skin, also the lung and the hair, and a few things that are inhabited very extensively with live bacteria. And this is not just me saying it, this is not just me showcasing it or advocating for it, but recently the World Economic Forum, who is tasked with devising the future of the world economy, wrote a whole report entitled Innovation with a Purpose, the Role of Technology Innovation in Accelerating Food Systems Transformation for a more inclusive, more sustainable, more nutritious, more efficient food supply chain, has identified CRISPR as a game changer. CRISPR is a breeding game changer for ag and is anticipated to be exploited in the next 10 years. It's almost the end of 2020 already, maybe a year that cannot end fast enough, but in the next 9 years and 1 month, right, maybe up to 100 million farms are anticipated and forecasted to use CRISPR-based genome editing to enhance farming. For more income, $100 billion extra of farming productivity. More yields, maybe 400 million tons extra. Less food waste, less food loss, and increased nutrition. Impacting over maybe 10% of the whole world population. Think about maybe almost 1 billion people benefiting from CRISPR-enhanced foods and crops in the next 10 years. But the challenge is, however good and promising and exciting the science is, we have to ensure the acceptance of science and technology by society. If you're curious about this, watch the movie. There's a lot of questions I hope we get to discuss in 10 minutes here. What is the ethics of editing humans, of editing crops, of editing livestock, of editing the world and trees in the forest? Are we going too fast? Maybe faster than we should? Or are we going too slow? There's people dying every day of genetic disease. And is this technology democratized enough to benefit all of humankind? Is it truly affordable? Is it truly scalable? Is it truly accessible? Is it truly equitable? How do we deal with the public lack of understanding of science? GMO technologies. People are scared of genetically modified organisms in our plate. How are they going to feel about genetically modified people? Like people moving around as genetically modified organisms who have had their genetic disease corrected. Are they GMOs? Are they safe? Are we biosecure? Having dinner with them, talking to them? Should they be incarcerated? Should they be, how do we manage with that, right? And then whose voices matter the most when it comes to acceptance, when it comes to guidance, when it comes to the dialogue about the regulatory paths for medicine, agriculture, and environment? How do we assess the risks? How do we assess the benefits? And whose opinions matter the most? That of scientists? Those of society? I mean, the Pope himself said, if we have the ability to address pain and suffering, don't we have a responsibility to use it? I asked a question which I hope we get to discuss. And this is why it's so important for everyone in the audience today to understand that however exciting and mesmerizing and powerful and tantalizing science may be, right? And occasionally the science provides great technology, right? The CRISPR science yielded CRISPR technology to use it for genome editing. But the true impact of science is hinging upon governments and industry and society accepting its use. And this is why it's so important to have projects like this, programs like this, outreach issues like this, not just to educate the next generation of scientists, but to understand the implications of doing science, the implications of being a scientist. As a matter of fact, besides my research programs, I teach at NC State. I teach a lot. So I do some research, I do some teaching, I'm speaking to a diverse audience at this point in time. I have a lot of business interest in the CRISPR space. People in my lab have to learn. I have to coach a lot of people in the industry on how to use it. I have to write grants and books and papers and not just science fiction, just science, right? And we have to advocate for science. And I'm hopeful that some of the people who are watching this right now are having a eureka moment. Being a scientist is not just doing research. Being a scientist is not just teaching. It's all those things. And doing those things well is important across the board to bring science and technology to society. And this is why I'm here. And this is why in 2016, a long time ago in CRISPR years, right, the Gardner Foundation awarded all those people here, right, 5 on the left and the 2 on the right, I'll get back to in a minute, for their work on CRISPR, bringing CRISPR science into CRISPR technology and applying that technology to enhance the world. Some of those people are very famous, right? So Jennifer Doudna just last month won the Nobel Prize in Chemistry. Amazing, an amazing, remarkable story, narrative, and person. But if you look right here, this gentleman right here, a little bit younger at the time, is the great Tony Fauci, facing a pandemic. That's what it takes, not just doing the science, but facing the pundits, facing a global health crisis, the politics, regulatory, clinical trials, the White House, public engagement, confidence in scientific enterprise, on and on and on and on. And sadly, earlier this year, we lost Dr. Frank Plummer, a pioneer in managing infectious disease. So there's many things you can do with science, especially today, to develop great solutions to the challenging problems that we have for the world, but also to take this to the next level. And I hope that you can see how CRISPR-based technologies have the potential today, have the potential tomorrow, have potential in the future to really enable the next generation of scientists to address the grand challenges that we have to face in food and ag, in medicine and biotechnology, to use the best science possible and the best technologies available to make the world a better place and solve all those problems. So I thank you all for the invitation. I'm trying to keep it short. It's 1:22 Eastern. It's perfect. And we have about 23 minutes for questions, and I'm sure there's many of them. So we'll just let Jesse manage the Q&A. Thank you.
Thank you so much, Dr. Barrangou. That was amazing. That was a lot of fun. The comments coming in on YouTube are fascinating already. So I'm really looking forward to this question period. We're going to start by going to Tuscaloosa, Alabama. Miss Foster's class, if you guys want to dive in and start off with a question, come on in and demute your mic and go for it. Go.
Is there any way your research can help find therapies or cures for GI diseases like ulcerative colitis and Crohn's disease?
Absolutely. Actually, ironically, we're actually working on this. So we are using those technologies to enhance probiotics, which are beneficial microbes that we ingest and colonize our gut to manage inflammation in our GI tract, to manage health and disease and digestibility in our GI tract. And there are some probiotics already on the market that are addressing UC and IBD. I believe you mentioned ulcerative colitis, inflammatory bowel disease, also IBS to some extent. And we're making them better so they can colonize better, help us digest better, and control the level of inflammation in our GI tract lining and epithelium, which is very important to not overreact to the outside world that is in our gut. So absolutely, and very much underway, ironically enough, in my lab. One of 200,000 labs using that.
Fantastic. Great first question, guys. All right, we're going to go to Miss Fowler's class, joining us for the very first time in the history of Exploring by the Seat of Your Pants for your talk. So I'm excited to have her in for a question, joining in Waterloo, Ontario. You got a question for us? Go for it.
Yeah, thank you so much for the opportunity. My class is going wild with questions, which is wonderful. Could CRISPR be used to help slow down, stop aging?
So yes, but aging is one of those multifactorial items for which there's not one magic solution, right? So we can't quite stop aging. We can slow down aging. There's a lot of people like my friend George Church who want to reverse aging and live to 150 years. And it's very hard at work to do that. But there's so many factors in place that yes, across the board, we are implementing CRISPR-based technologies to correct genetic diseases, to fight off infectious diseases, to enhance the microbiomes with which we're affiliated, to make healthier, safer, more sustainable foods, better air. There's also uses of CRISPR outside of genome editing for therapeutics, whereby we can use CRISPR for diagnostics, so we can diagnose people better, faster, and more affordably. And we can go into maybe futuristic approaches where we can grow organs and tissues for organ donation, not relying on organ donors, but relying on the lab. And I would compel your class to watch Human Nature. There's a segment in there on humanizing pigs, so we can grow human tissues in them and enable us to address diseases that would require otherwise organs to be lifted from other patients or donors. So Human Nature, it's on Netflix. I think it's on Netflix throughout the world. I know it's in the US and has been in Europe. I hope it's in Canada. Watch it. It's great. It's very informative, and it makes for a great science session in a science class, and assuredly will prompt a lot of questions.
Fantastic. A quick note, you mentioned George Church, and he was in that Human Nature documentary, so I'd encourage every class to check that out. George has also written a whole bunch of books. We've been getting questions on YouTube about where people can learn more. So we mentioned George Church. Jennifer Doudna, you've mentioned numerous times. She has a book called A Crack in Creation, which is a really fantastic read. And then, of course, check out Gairdner.org and at the Gairdner Award to find out more about these amazing people doing great medical health and global health research around the world. So really lots of ways to keep learning going, and I will share all this information with the classes afterwards in an email.
Yeah, lots of books, including Kevin Davis's book that just came out the day after the Nobel came out, Editing Humanity. So you can't go wrong with any CRISPR book.
Fantastic. Awesome, guys. All right, let's go to Notre Dame High School in Belmont, California. Ms. Gerard, if you've got a question for us, come on in.
Yes, thank you so much for this talk. It is really sparking a lot of questions. And one of the questions a student has is that CRISPR seems to act on a cell-by-cell basis. So how is it possible that you can alter like a whole human organism if you're doing it one cell at a time? How does that work?
Yeah.
So that's a great question. And right now, we do not have a solution for systemic altering every single cell in a whole patient. So the first generation therapies are focusing on delivery modalities whereby by impacting enough cells, you have a therapeutic impact. So think blood diseases, we can inject a few cells into the blood and they can multiply thereafter. Think liver diseases, eye diseases, muscle diseases, maybe even kidney diseases, and some long delivery modalities that enable us to impact enough cells where we have a therapeutic benefit. But if you want to target something like HIV, we're not yet at the point where 100% of the cells in 100% of a patient can efficiently remove a virus 100% of the time. So we're not there yet. We may even never gonna get there. There's a lot of discussions about engineering the next generation of people by altering oocytes and altering the germline. At this point in time, the National Academy of Sciences, the Royal Society, and the academic community at large have all agreed upon the fact that we're not ready to do that at this point in time. And even if we wanted to, we couldn't, because the technology is not mature enough, advanced enough, safe enough, and enhanced enough to do so. So great question, great challenge for sure. We're not there yet, but we do not need to be there in order to have a therapeutic benefit. So the first generation, likely the second generation, even the third generation of therapies will focus on partial impacting a subset of the cells so we can still have a therapeutic benefit.
Yeah. This is such a unique talk. Again, we've got some new teachers coming in today, and I love this because this touches on so many ethical issues that so many of our other presentations don't. When you're with sloths in Costa Rica, as cool as that is, there is no ethical dilemma. The sloths are adorable, but I love that we get to cover some really deep topics here today. And again, the YouTube comments are amazing. We're going to get to some of those in just a minute. What I want to go to now is Miss Wafer's class, the Laurel Springs School. So they've got students all over the world. So Miss Wafer, if you want to come on in, go for it.
Awesome, thank you. Yeah, we're a virtual school. We've been around for 30 years. And I today sent the invitation to our Scrubs Club, our Model United Nations Club, our Debate Club members, and our Environmental Club members. So some of our questions are gonna focus in on those kinds of topics, and any information that you could give us to utilize in those clubs would be helpful. So the first question is maybe some examples, it's connected to the environmental club, some examples of how gene editing can be made to make agriculture more sustainable.
Oh, it's a great question, and definitely a focus that is very close to my heart and appetite and business and scientific interests. So I'm going to give you two examples, right? The one from the World Economic Forum to provide more sustainable agriculture and increasing yield beyond what we can do right now with breeding programs, fighting off diseases, better exploitation of limited water supply chain and the like. So we can go into crops of interest like corn and soy, and many others like rice, right? And use CRISPR to accelerate the breeding process, facilitate the breeding process, multiplex the breeding process, and alter every single gene that we knew is associated with a benefit. A yield benefit, a pest resistance benefit, a disease benefit, a water usage benefit, a drought resistance benefit, any combination thereof. We can do that easier, faster, and more affordably than we ever have. And rather than take the time to breed and crossbreed, and sometimes there's genetic drag that comes with it over years of time that it takes to breed in and breed out. I use CRISPR to surgically do it better, faster, and easier. Now, the bigger issue besides food and ag to me is sustainability. Global warming is a major challenge, right? Climate change is a major issue. And I, in a partnership with my friend Jack Wang, have been working for years on using CRISPR in trees to breed a better forest. So can we make the forest industry more sustainable? A major product of the forestry and the forest industry is pulping. So can we engineer the lignin content in trees to make the processing more efficient, less toxic, less demanding, less taxing, and less chemical dependent? So there's a lot of things to do. There's hundreds and hundreds of traits we can go after. There's thousands of genes we could go after. But across food, across ag, I mentioned crops, but it's the same that happens to livestock, the same happens to trees, the same happens to ornamentals, the same happens to all ecosystems in which living organisms can be enhanced and bred. It's broadly applicable, but with timelines for trees, right? A tree you plant today may not be harvested for 16 plus years. We have to start now, given the pace at which climate change is unraveling and occurring before our eyes. So I'm looking forward to the next generation of scientists listening in today, helping me and others tackle those problems, because we've got a lot of issues to deal with. And I'm sure some of you are motivated enough and aspirational enough and bold enough and visionary enough to join Team CRISPR here to address those issues. Thank you.
You know, if for any of our students watching today live or on YouTube, if you find something in your life you're as passionate about as Dr. Barrangou is about CRISPR and its potential, you're set. Like that's what you're aiming for in life and whatever your field may be. I love that you talked about something that I think is really integral for all our classes to understand, which is that whether it's agriculture or a lot of these things that CRISPR could be potentially used for, we've been doing these sort of modifications for decades and sometimes in hundreds of years. CRISPR just presents us with the opportunity to do it in a much more targeted, easy, faster, and better fashion where it's not so haphazard with our slow changes. I mean, you look at agriculture and it's a long history of mucking around and hopefully finding something that fits, and CRISPR gives us the tools to do that in a much more efficient and effective way. So I'm really glad you covered that answer. Julia and Victoria, let's head to Vancouver. If you guys have a question for us, come on in, demute your mic, and go for it, ladies.
Hi. So you mentioned that you find the gene to edit, kind of like Control F in a document, and I was wondering, how exactly do you find it? What's the method by finding it? Because I know there's sort of so many genes and base pairs. How do you find the specific one that you want to edit?
So very good question. So think to be or not to be, that is the question, right? You have to have a short string, a series of letters that is unique in Shakespeare's comprehensive works, right? Thousands of pages, right? So the way CRISPR works, it's a combination of two things. It's an enzyme called typically Cas9, there's others, we'll just stick to Cas9, that has the ability to go somewhere and cut DNA. That's one piece. And the other piece is what's called a guide RNA. So think the Control F function and think the string of text you put in. So the guide RNA is encoding in an RNA sequence, which is what won the Nobel Prize in Chemistry in 2020, 20 letters in a row. a combination of A, T, C's, and G's that is 20 letters long that will specify where Cas9 is going to go and generate cleavage. And that cleavage occurs precisely 3 letters away from the 3' end of that sequence. So it's a ruler anchored mechanism that will control F, those 20 nucleotides in the genome, scan for them, find them, bind to them, create an R loop if you're curious and scientific, and then the dual undernuclease system is going to count 3 letters from the edge, -1, -2, -3, and cut DNA precisely there. That's how it works. That's how it functions. If you're curious about this, go to the nobelprize.org website. Don't quote me on that, but I think it's correct. And they will explain that. Or go listen to the great Jennifer Doudna's introduction to CRISPR on YouTube. I'm sure there's a bunch of videos that are there for all kinds of audiences that have different layers of complexity and technical details that explain how that works. But this is an endonuclease that is programmable and looks for a 20 nucleotide long query in Control F and cut.
That was a very good attempt to explain that in about a minute in a broadcast. But yes, there's lots of great visualizations. It's something that very lends itself to visual thinking. So if you guys want to check out books, articles, TED Talks, there's lots of resources out there to understand a little bit more about that process. So great question, ladies. All right, we're going to take a couple questions from YouTube, and then we're going to do a second round with all our live classes. So there's been a lot of questions on this line, like the one I'm about to bring into the broadcast by Venetia. So could companies try and profit off this, do things that, maybe aren't in the public interest or a little bit odd, or in this case, talking about creating a perfect child, so to speak. So this is a big concern that a lot of people have. I'm sure you've heard this question many times. So if you want to answer sort of the potentials and dangers of this technology, that would be great.
Yeah, so technology is neither good nor bad, right? It's how it's being used and who's using it. So as I mentioned, imagination may be our only limitation. And with 2 million scientists doing it all day long, right? You can imagine indeed that even a small percentage of those people, even 1 in a million, having bad motivations or being coerced into doing something stupid or bad can happen. And we had that happen. Zhenghui He is a scientist in China that 2 years ago allegedly used CRISPR to edit 2 twins that were born without a gene called CCR5 to make them resistant to HIV. And the way he did it, the way he went about doing it, the tools that he used, the ethical shortcomings, the disclosures, the risk for the patients, and on and on and on, there was a lot of objectionable things to that effect. So the scientific community at large has been engaged in hundreds of meetings and conversations, including the public, including regulators, including scientists, including religion, thought leaders, and the like, all minorities and all majorities across the world, to try to understand what the risks are. The upside is great, the promise is great, the potential is tremendous, but we have a responsibility to use those tools wisely and cautiously. So there are indeed risks, there are indeed potential for abuses, to breed the next generation of enhanced humans, or select what genotypes you want or not. Again, watch the movie. There's a great Gattaca segment and Jurassic Park segment in there addressing this. So there are risks for sure, but we are aware of them, mindful of them, and we're embracing those opportunities to discuss what the boundaries should be for the exploitation, commercially and scientifically, of those very powerful technologies across the globe, so we can be responsible, so we can be safe, and so we can ensure and guarantee that the best potential use of those powerful technologies is what we're focusing on. Because unless we do that, there will be abuse. But besides the potential abuse, there will be rejection of science by the public. There will be mistrust in science and scientists by the public, and we cannot afford to make that mistake. So I could not be more excited about this question. It's a very important contextual item that needs to be accounted for, discussed openly, discussed extensively, thought about by thought leaders across the world and across denominations and across biases and lines of professional responsibilities. And it is happening in real time, and it's extremely exciting.
That is a very, very thoughtful answer, and I really appreciate the question. So thank you to our students pouring in on YouTube. Just a quick note, you mentioned Gattaca and Jurassic Park. These are two, I mean, they're decades old now, and they're still probably the best two sci-fi movies ever made on this topic. So I'd encourage everyone to check them out. I think most people have seen Jurassic Park. Gattaca is another amazing film. Lots of really interesting stories and ideas bandied about in it. Dr. Barrangou, do you have time for another 5 questions with our live class?
Of course.
Awesome. Well, let's go to Miss Foster's class. If you guys have a second one, just demute your mic. Come on back in.
What type of educational background would you need to work in this field?
Yeah.
Very good question. So any, as a matter of fact, you can do a science communication major, right? You can do ag, you can do food, you can do therapeutics, you can be a vet, you can be an MD, you can be an engineer, you can be a bioinformatician, you can be a big data analyst, you can be a business major, right? So I went to grad school a number of times. I went to engineering school, I went to business school, I got a degree in genetics, a PhD in genetics. So there's a lot of different disciplines that are applicable to this, even physics, right? Even chemistry. I mean, so almost any scientific discipline can be somewhat applicable to genome editing and genetics in general. And depending on the field that you're interested in, the kinds of organisms you're interested in, the kind of applications you're interested in, there's almost no limit to of the major you will choose to do so. Now, obviously, if you want to do genetics and cure genetic disease, being an MD, PhD with clinical access and encyclopedic knowledge about genome editing in humans is great, but it's not a must, right? You can be a zoologist, you can be a mathematician, you can be an engineer, you can be a climate change specialist, you can be a meteorologist, you can be an astronaut to do CRISPR experiments in space like it's happened with NASA before. So there's almost no limit whatsoever. You don't have to be a scientist. You can be a social scientist and think about psychology, think about communication, think about marketing, think about regulation. We have a lawyer on the editorial board of the CRISPR Journal who doesn't have scientific training because there are legal ramifications to it. So there's no limitation whatsoever, but more biologically, medically relevant items probably is a preferred path.
You could be a butcher, a baker, a candlestick maker. In all of our sessions when we have NASA scientists and whatnot, I love this message that no matter what field you're specifically interested in, if you want to get involved in some of these great scientific challenges and fields, there's ways to get involved through a huge variety of careers. But you single-handedly had the most examples of careers ever in one of our broadcasts. So thank you so much for that.
Ask for the record for that.
The award, I'll send it in the mail when we're done. Ms. Fowler's class, you had a great question in chat, but I'd love if you'd share it live with us. So come on back in and go for it.
Yeah, a lot of my students are asking questions about things that go wrong and problems. Do those types of things get published as well as the successes?
Absolutely. It is very important for science transparency, scientific trust in the enterprise, the process, and the community to do so. There's been a lot of papers published about the efficiency of CRISPR, the inefficiency of CRISPR, the specificity of CRISPR, the misuse and abuse of CRISPR, the overpromise and underdeliver, the mosaicism, the off-targeting. So absolutely, it is important. It is part of the scientific method. And again, it's part of the process that we have to all go through to build trust in the system. And right now, we're very excited about the early results for clinical studies, clinical trials, clinical endpoints early on. But until we go through using those therapeutics to treat thousands and thousands of diverse people in phase 3 trials, we still won't know how ubiquitously safe and impactful those modalities will be. But it's very much part of the process. It's very much part of the standards. It's very much part of the onus that we all have as scientists. And we have a duty to ensure that not just the upside, but the downside, the downsides, the risks, the liabilities, the challenges, the problems, the issues, the misuses and abuses are maybe not equally covered because we don't waste half the resources on this, but we're going to spend some of our resources on this and invest some of our resources on this to ensure that they are safe and that the risks are measured. The great Tony Fauci, again, I mentioned him a couple of times, is a great example of this. We have to go as fast as science enables us to do so, but only so fast. And we have to understand and appreciate the fact that responsible scientists ensure that what we do is not just efficacious, but also safe. Because if it's not safe, we will know and we'll act on it. So that's very much part of the process, very much part of the method, very much part of the focus. And we will, and will continue forever to ensure that we cover the liabilities and the risks as much as the upside, for sure.
Yeah, I'm really, really happy we're getting very into this question throughout the broadcast today. And Dr. Barrangou is a perfect example of it personally, but as someone who's followed the history of science for quite some time, genetic research is one that is incredibly transparent, incredibly open for exactly this reason. It's something that invites so much skepticism, so much concern and ethical considerations that the scientists basically since the 1960s and 1970s have really organized themselves together and worked very openly with governments, with the public, with science communication. So that's why there's such an abundance of amazing resources out there to cover. If you're keen to learn more about this topic, there's some really, really great stuff. So I'm thrilled we're getting this question so much, and I'm excited to keep going with our Q&A right now. So let's head to Ms. Gerard's class, back to California. Come on back in and go for it.
All right, so we've got a couple of questions. And we're going to kind of put them together. The question is really about how CRISPR-Cas9 may be used currently to develop therapeutics for COVID-19. And if and when that becomes successful, how available and how affordable will those treatments be? Another social justice ethics kind of question there.
Yeah, very important. So equity is very important. Affordability is very important. That's one of the great things about CRISPR, and you have time to discuss because I want to cover so many things. You can only go in depth so much. But one thing that has changed fundamentally for CRISPR is it has democratized the researchers and provided access to a very powerful, portable, efficient, affordable tool. And those benefits that we see on the research side will carry over to the therapeutic side. That being said, a gene therapy is still very expensive, sometimes even cost prohibitive. So making that available is one thing, making that affordable is another, making that affordable to all is yet at the third level. So we're not there yet, but we're getting there. We're making a lot of progress. There's a lot of investments in this space for sure. And this is an example for why CRISPR technologies and fundamental research investments are worth it in the end, because when you have a breakthrough like this, you can lower the cost therapeutically. Antivirals are tough. Like I mentioned before, for HIV, we don't have yet the ability to efficiently remove 100% of the whole tissue, let alone the whole individual viruses. So in the case of COVID-19, what the opportunity that we have compared to other viruses and infectious diseases is that it's localized to some extent, primarily to the lung. So we have some delivery modalities for the lung with aerosols that are in play. They're difficult, they're challenging. Efficacy in terms of percent lung cells that get delivered to is tough, let alone the editing outcome and the removal of COVID-19, coronavirus V2. But there's a path there. There's some early proof of concept studies in labs, including a friend of mine, Stanley Chi at Stanford, who have provided proof of concept that you can use CRISPR to target COVID-19 RNA. But it's very early, it's very preliminary. And as a matter of fact, the primary benefit thus far, in my opinion, of the CRISPR community on the COVID-19 pandemic is diagnostics. So Jennifer Enfong, Mammoth, and Sherlock have both developed kits that are very affordable, paper-based tests in partnership with big pharma companies that are democratizing and making affordable and scalable testing. So we're not quite there yet, but we're on the cusp of very large-scale manufacturing of CRISPR-based COVID-19 testing that will enable us to test before we have a therapeutic. So the therapeutic's not there yet. The Moderna news, Derek Rossi is a Moderna founder, a friend of mine. There's exciting news from Moderna, there's exciting news from Pfizer and Johnson & Johnson and others. So I think the vaccine modality is there. And of course, there's a lot of people using CRISPR to generate vaccines as well, more prophylactic than a therapeutic. So to answer your question about therapeutics, we're not there yet. Vaccines, we're going to get there. Testing, I think we're on the cusp of being able to do diagnostics. So the CRISPR community has risen to the challenge. They've embraced the challenges, let alone doing the testing in their labs. Again, Jennifer has done that in her own lab at UC Berkeley at the IGI. And others have in New York as well, and other places, Sam Sternberg at Columbia and others. So it's very important to make ourselves available to tackle those kind of challenges and issues as they arise. But to answer your question, honestly, we're not there yet for antivirals. We're not quite there yet, but we're on our way for vaccines. And the biggest impact so far has been testing, which is not trivial whatsoever. Even almost a year in, still surprisingly, perplexingly a big challenge. But as I said in the recent editorial, we all need a moment of science. And science will work our way out of the pandemic. And there's a lot of reasons to be excited right now with the news this week and with forthcoming news about addressing various aspects of the pandemic, not just antivirals, but I think vaccines, diagnostics, number 1 and 2.
I knew we were going to get that question. I'm really glad we did. And again, the Moderna news, really, really exciting. If you haven't seen this yet, it's in the last couple of days, really, really exciting. Multiple vaccine candidates, which is fantastic, and more to come down the pipeline. What was I going to say? A quick note for everyone. Oh, dear. Completely forgotten. Oh, if people are keen, I mentioned this at the beginning, Dr. Tanya Watts last week on COVID and vaccines. If you want to check out that talk, it's on our YouTube channel, part of our Gardner Foundation series. So I'd really encourage you to do that. A nice follow-up to this chat today with Dr. Barrangou. All right, we're going to take 2 more questions. We could go all day. There's been so many questions. Miss Waver's class, come on in, and then we'll go to Julie and Victoria to wrap up in a minute.
Awesome. Well, I'm so glad you mentioned NASA, because I did not think of sending this to our astronomy club members, but Sam is in 10th grade in Maryland, and the question is, could plants be adapted to soil and atmospheric conditions found on Mars?
So yes, I mean, I don't know how much we actually know about the granularity and details of farming on Mars at this very point in time. I know my friend George Church did help check some numbers in The Martian when that was produced and written. I think there's hope. There's a lot of reasons to be excited. Synthetic foods, impossible foods, and genome editing certainly is a very timely opportunity. So absolutely, I think in this case, microgravity or no gravity leaf formation and capturing rays, like UV rays and sunlight. So I know there's some work being done in corn on leaf angle to really control maximal exposure and capture of UV rays. So I think genome editing there can have a direct impact on enhancing this. I think microgravity and water usage are 2 other traits that would be critical and important. And presumably, when we get into space, we'll have much higher control of infectious disease and viruses and pests. So I hope we don't have to spend so much time addressing those challenges of infectious disease and focus more on the traits of importance for transforming energy into valuable chemicals to the plant.
Awesome, guys. All right. One last question to wrap us up. Again, it's worth going overtime with this because there's so much to discuss and so many more things I'll share with email with all our classes again that registered today. So we'll go to Vancouver for one last question. Ladies, take us away. Wrap us up.
So I'm interested in what kind of potential mental health side effects there could be for a designer baby who would maybe grow up and find out that their parents changed them. And would there be any sort of safeguards in place to protect that?
Big question.
It's a great question. And it is a great question tremendously covered in Human Nature. So I'm not going to give you guys the ending because it would really not do justice to the movie, but the wisdom of David Sanchez when he pretty much answered that question posed to him, and listening to a 13-year-old be philosophical, reasonable, and wise enough to provide the best answer possible to this out of the voice of a patient, a child in some way, at least legally a child, right? But there's a wisdom, clearly not a child. Watch the movie, think about it, talk about it with your friends, ponder it personally, philosophically, technically, ethically, medically. And being a parent is tough, but being a patient or a parent of a patient child is maybe even tougher. There's a lot of complexities and layers intertwined there with regards to what would you do if, and having people ponder the question, arbitrarily about a thought process versus being emotionally loaded when they answer that question is very interesting exercise to go through for people who are afflicted by disease or other combinations thereof. Watch the movie, call me when you've seen it, and we'll discuss that again. Thank you.
We're going to put Dr. Barrangou's number in the email at the end. No, it is worth looking into, guys. You spent, I promise not. We spent an hour today talking about this. Spend that extra 90 minutes. If you don't have Netflix already, it might be worth getting it just for this. These are some really, really big discussions. We're starting to have these as a society now. And over the next 10, 20 years to come, there'll be many more opportunities to dive into this topic in a huge variety of fields, whether that's space, agriculture, mental health, all these amazing things. So I would really encourage you to check that out. And again, I'll share some nice book ideas too if you're more keen on that. A nice segue with mental health. I do want to note that next week on Monday, 9 in the morning Eastern time, we have Dr. Vikram Patel. He is a Harvard researcher. He's going to be talking about cutting edge mental health research. So if you want to follow up with that as a nice follow up to this, I continue on with our Gardner series. I think it'll be a fantastic presentation as well. Do check out Gardner.org and at Gardner Awards for more on the amazing organization, all the awards that they give to some really, really top-notch people in research around the globe. And what I want to do now, Dr. Barrangou, is what we do at the end of every broadcast. I'm going to bring in all our live groups. So in Alabama and Ontario, California, Miss Waivers, Worldwide Classroom, and BC, say a huge thank you for all that you share with us today. So everyone, you guys are all in. Thank you so much.
Yay, thank you.